We are continuing on with the Chapter 8 lecture notes. We left off by looking at the structure of a typical multipolar neuron. I showed you some areas called Niesel bodies, which are important in forming the gray matter of the central nervous system. Niesel bodies consist of free ribosomes and rough ER. And that is something that is like a characteristic of a neuron versus, other type of cells. And then we have the axon hillock. The axon is just the extension from the neuron cell body that takes carries the electrical impulse to a target. The axon hillock is the actual point on the axon. It's where the axon begins, and it's the point that the action potential or electrical impulse or nerve impulse is generated. As far as the regeneration, the ability of neurons to regenerate if they're damaged or destroyed, is that most central nervous system neurons cannot divide. You may have heard different times over your life, especially in elementary school and middle school and high school, and during your public school education, that when you drink alcohol or do drugs, you're destroying your brain cells. And it's maybe oversimplification, but it's true. And when they're gone, they're gone. Now you can, once you've destroyed neurons, you have so many that you can still do many of the same things, because certain activities will just learn a new pathway and use different pathways. That's still, you can have nerve damage and you can be rehabilitated from it, but you can't just, once a neuron is dead, you can't just make a new one. It's once it's destroyed, it's gone, and it's not going to be replaced. So that's important to know about your nervous system. It's important to protect it. So multipolar neurons are the only ones I've highlighted on here. There are other types, unipolar and bipolar, but the multipolar neurons will have two or more dendrites extending out from the cell body and then a single axon. All of the motor neurons are multipolar neurons, and all of the neurons within the central nervous system are multipolar neurons. So there are other neurons and other types. We're going to we're just going to learn that one type. Just like we had a sensory division of the nervous system, we had a motor division, and we also have a central nervous system. We also can divide neurons up this way. There are neurons that function as sensory neurons. They receive information from sensory receptors. And motor neurons carry instructions away from the central nervous system. So sensory neurons will receive information and relay that information to the central nervous system, whereas motor neurons are going to carry information away from the brain and the spinal cord. Interneurons are the ones that are found entirely within the brain and the spinal cord, and they're going to basically receive the sensory information and decide what to do with it. Decide which motor neurons to stimulate and the signals to send out, from the central nervous system. All right, for the sensory neurons, we have two types of receptors. We have the somatic receptors and the visceral receptors. So for somatic receptors, we have external receptors that monitor the external environment. So we have receptors, a lot of these are in our skin, but not all. Some are in our special senses. We have ones that monitor pain, touch, pressure, and even the sensation of hot and cold in the skin. We have vision receptors in our eyes, hearing receptors, and balance, equilibrium receptors in our ears. And then we have taste and smell receptors. we have proprioceptors that monitor the position and movement of the body. Some of those are located within our skeletal muscles and our joints. And we have visceral receptors that monitor the activities of internal organs. They provide sensations of distension, like when your stomach is full or your bladder is full, deep pressure and pain. And then we also have, it also monitors things that we don't notice the sensation of as directly, like, say, for example, the levels of certain chemicals in our blood calcium levels, blood oxygen levels, blood carbon dioxide levels, blood glucose levels, blood pressure, so those are going to be monitored by visceral receptors. And then for motor neurons, we have somatic motor neurons, and these target the skeletal muscle, and we call those in the neuromuscular junction, we just called it a motor neuron that innervates the skeletal muscle. And then visceral motor neurons innervate either cardiac muscle, smooth muscle, or glands. Interneurons are by far the most numerous type. There are about 20 billion, I said millions earlier, I should say billions of neurons. We have about 20 billion interneurons located entirely in our brain and our spinal cord. And they play a role in higher functions like memory, planning, learning, coordinating the motor response based on the sensory input. So this is all done by the interneurons. Now we're going to move on to the cells of the neuroglia. Neuroglial cells help to support and to nourish and support and just have various functions to keep our neurons healthy. There are 4 neuroglial cells that are found within the central nervous system. They're called astrocytes, oligodendrocytes. I'm going to say that one again. Oligodendrocytes, microglia, and ependymal cells. That word again is ependymal. Then there are 2 types that are found within the peripheral nervous system. They're called satellite cells and Schwann cells. So let me blow this up. This is just a picture of the central nervous system. That's actually the spinal cord, and it's showing you the different neuroglial cells. So we have some neurons here. This is an axon covered with myelin, which is like insulation, and myelin is a very fatty tissue, and it's white in color. And in the central nervous system, it is produced by oligodendrocytes. So here's an oligodendrocyte that's forming this tissue that wraps around the axon of a neuron, forms the myelin that wraps around the axon of a neuron. So oligodendrocytes form myelin sheaths. This is called a myelin sheath. They produce myelin sheaths in the CNS. Then we have these cells called astrocytes. They have extensions that they connect to the neuron, and they connect to our blood vessels. And what they do is separate the blood from the neurons. It's in the brain. This is called the blood-brain barrier. But they form the same function in the spinal cord. We don't want our blood contacting neurons, because the blood can carry wonderful things, but also the blood carries waste products, it carries toxins, it carries pathogens that cause disease. And, knowing that when we lose neurons, we can't replace them, we have to protect them from all that stuff. So the astrocytes form the blood-brain barrier in the brain, and then they kind of have a similar function with the neurons in the spinal cord. Microglial cells are phagocytes, so they're going to function in defense, engulfing pathogens and destroying them. Ependymal cells produce some of the cerebrospinal fluid, and they line fluid-filled passageways. These are ependymal cells right here. But they produce some of the cerebrospinal fluid, or CSF, and then some of that fluid is produced by the choroid plexus, which we'll learn about later. But anyway, they help circulate that fluid. So that would be the ependymal cells. Astrocytes, again, maintain the blood-brain barrier. Oligodendrocytes produce myelin. Now we can talk about what the difference is between white matter and gray matter. White matter are areas dominated by myelinated axons, and gray matter is areas that contain neuron cell bodies, because remember, in the cell bodies we have Niesel bodies, which contribute to the gray color, and dendrites and unmyelinated axons. All of that is found in gray matter. So oligodendrocytes produce myelin sheaths in the CNS. Microglia are phagocytic cells derived from white blood cells, so they function in defense. And ependymal cells produce and circulate cerebrospinal fluid. And in addition, the choroid plexus produces cerebrospinal fluid. In fact, the choroid plexus, which is just a massive a capillary bed, the choroid plexus produces most of this cerebrospinal fluid, but then the ependymal cells produce some, and they really are involved in circulating that fluid. Schwann cells of the PNS function like oligodendrocytes in the CNS. So the Schwann cells form the myelin sheaths on the axons in the peripheral nervous system. And all axons in the PNS are covered with myelin. Satellite cells in the PNS have a similar function as astrocytes in the CNS. They keep the nerves from being coming in contact with blood. Now, you might hear the term ganglia. In the peripheral nervous system, ganglia would be collections of neuron cell bodies surrounded by satellite cells. And then nerves are bundles of axons. Some of those nerves can be motor and some are sensory, so that would be a mixed nerve. But when we talk about a nerve, that's defined as bundles of axons. And we have two types of nerves. Spinal nerves originate from the spinal cord, and cranial nerves originate from the brain. So we will learn the cranial nerves in detail. We'll talk about the spinal nerves, but we'll go in detail with the cranial nerves. Now, we're going to go on to the checkpoint questions. Which type of glial cell would increase in number in the brain tissue of a person with a central nervous system, CNS, infection? So that's going to be the microglia. And then in the peripheral nervous system, neuron cell bodies are located in ganglia, goes in the first blank, and surrounded by neuroglial cells called satellite cells. Satellite goes in the second blank. All right, we're going to go on and talk about the membrane potential of a neuron. We're going to focus on three types of membrane potentials. The resting membrane potential, the graded or local potential, and what's called the action potential. So you need to understand what potential means. The membrane potential is the voltage across the plasma membrane of the neuron. It's measured in millivolts, just like voltage in when we're talking about electricity. So the resting membrane potential or voltage of a neuron is negative 70 millivolts, and that negative is the inside of the plasma membrane. So it's just letting you know that the inside is negative relative to the outside. The outside is got a carries a positive charge and the inside of the plasma membrane carries a negative charge. And if you hooked up a voltmeter to the plasma membrane, one end measuring the inside of the plasma membrane and one end measuring the outside, you would get negative 70 millivolts on the voltmeter. Now, what causes this resting membrane potential? Some different things. I'm not going to make sure that you understand every single part of this, but some things are important. It is important to know that the extracellular fluid, so outside the cell, outside the neuron, is high in sodium and chloride ions. And the intracellular fluid is high in potassium and negatively charged proteins. And those negatively charged proteins can't leave the cell. So that really contributes a lot to the negative charge on the inside of a resting neuron, one that's not conducting an action potential. But what actually maintains the resting membrane potential is something called the sodium-potassium exchange pump. So really that's what I would want you to know about this negative 70 resting membrane potential. Know that the resting membrane potential is negative 70. Know that what that means is the inside of the cell is negative, the outside is positive, and that the resting membrane potential is maintained by the sodium-potassium pump. And we'll go into more detail with that in the next recording.